Testing. Test. Okay. Testing my mic. Okay, I think mic is okay. Slide okay. Camera okay. Okay, now it's already 8 o'clock. Okay, so we will start our class now. Okay, so today class is the additional extra class to replace the last week class, because last week I due to my university event, so I cancel the last week class. So for today, actually I will finish up the chapter 12 lah, chapter immunity. So as you guys know that yesterday we already covered up the first two topic, right? Immune system and also the immunity development. Okay, so for today we will continue to the antigen antibody reaction, which is still under the immunity development. There's left one topic, and then after that we will proceed to the concept self and non-self, and also the autoimmune disease, immune disorder. Okay, so antigen antibody reaction, I think this one is only the topic that actually you learn from SPM lah. So when you heard about this term, I think everyone are familiar with it. Agglutination, neutralization, precipitation, right? And also phagocytosis. Yeah, this one is that you learn in SPM. So I think it is only the topic that you learn in SPM lah. But in STPM, we will add more substance. The mechanism we call as activation of complement system. Complement activation. So neutralization, agglutination, and precipitation are totally same that you learn in SPM. And at the end of the reaction, they will trigger phagocytosis. Yeah. Okay. So for neutralization, so the verb already say neutralize. So it is the antibody will neutralize your virus, your pathogen. So meaning that your antibody will bind to your antigen, so that this antigen cannot bind to the host cell. It cannot infect your host cells anymore. Once your antibody already bind to the pathogen, then this antigen cannot bind to the host cell, meaning that this bacteria or virus cannot bind toward the host cell. So this process is we call as neutralization. So when your antibody bind to the pathogen and antigen and interact with the host protein, so masking them and inhibit them, all the pathogen into the host cell. So the pathogen like bacteria, virus or fungi cannot infect your cell lah. Okay. So antigen pathogen complex are then eliminated often after phagocytosis by macrophage. After that, this complex will be eliminated by phagocytosis lah. Okay, so the neutralization main role is to prevent the bacteria, the pathogen or virus to enter into the host cell. And then another one, agglutination. So agglutination is like a clamping process. The antibody will bind toward the antigen. They still bind toward the antigen on the surface of the bacteria, and then it will aggregate all the pathogen at the one place. It will aggregate it. Dia akan kumpulkan semua bacteria tu dalam satu tempat. You will know that bacteria is organism too, so it can freely move one. So this antibody, dia like macam polis lah, tangkap semua bacteria tu kepada satu tempat, and then after that this large complex, the later will be phagocytized by macrophage. So macrophage won't be easy to engulf this bacteria. So compared to macrophage engulf one by one. So if they are packed together, they are huge complex like this, macrophage are very easily to engulf them. So this is what we call as agglutination. It claim the antibody, clump many bacteria into one place. Another one is precipitation. So precipitation is also similar to agglutination. The difference is they bind to soluble antigen molecule. The agglutination is bind toward the antigen, or you can toward the antigen with on the surface of the bacteria. But precipitation is by the soluble antigen, meaning that this antigen is flow surrounding one. It does not on the surface on the pathogen one. It is freely moving surrounding. So this antibody, you can see the white shape is antibody, yellow color is antigen. So the antibody will also bind to it and aggregate them into one places, so that macrophage is easily to engulf them lah. Okay, another one is the new thing is that complement fixation. What is another name is we call as activation of complement system, which involve your complement protein. So you see this diagram. So this is your complement protein. This is another protein, and that antibody is here, and then this is your antigen. So this one is special when the cells are already, your host cells is already infected by the cell. You still remember cell-mediated immune response is totally destroy the cell, right? Totally destroy the cell through the cytotoxicity cell, and cytotoxicity cell will secrete the perforin and also granzyme to destroy the cell, right? So in human immune response, your antibody also can trigger the complement fixation or trigger the complement fixation or activation of the complement system, also can destroy the cells. The main function is, the main effect is still same as the cell-mediated immune response. It destroy the cell, but the way they destroy are different. This one is use antibody. So this not only antibody, it require also the complement protein. So this antibody will work together with the protein of the complement system. So firstly, your antibody will bind to the antigen on the surface of the foreign cell to activate the complement system. So foreign cell, so normally each cell can be bacteria cell, and can be fungi cell, and can be virus, any cell, foreign cell, any pathogen I can say. Okay. So this antibody will bind to the antigen, and then it will activate the complement system. So antibody at the same time will also bind toward the complement system. So this will trigger the formation of your membrane attack complex. So it will form the membrane attack complex. So this one, membrane attack complex, will bind toward your plasma membrane, the plasma membrane of the pathogen. So it will form the pore in the plasma membrane of the cell. Hence, the water and ion will rush into the cell, causing it swell, and also lyse. So it will cause lysis. So it will produce the factor that promote inflammation or stimulate the phagocytosis. Yeah, so the key role is complement protein and antibody. So firstly, your antibody need to bind to the antigen, and then at the same time, your antibody also can bind to the complement protein. So once they already bind to it, this complement protein will form the membrane attack complex. So already say a membrane attack complex, so it will attack your plasma membrane of the cell. So when it bind to the membrane, bind to the plasma membrane, it will form the pore. Satu lubang. It will form the pore. So since there are the pore, so all the water molecule and ion outside the cell will rush into the cell, so that your cell now is highly hypotonic, and it will die lah. Okay, so this is about the complement fixation. Okay, so that's all about the antigen antibody interaction. Okay, next we go to concept self and non-self, the third topic of this chapter. Okay, so you need to know that why you want to learn about the concept self and non-self is you need to, if you still remember yesterday about the topic of the development of B cell and also T cell, you know that B cell and T cell only should be in theoretical should be attack the foreign molecule only, foreign antigen only, right? In theoretically, so you in doing the development of B cell and T cell so that they will undergo the negative selection, which ensure that the B cell and T cell only can attack the foreign antigen only. It will not attack our own antigen. You need to know that in our bodies, we also containing antigen. Our self antigen produced by the cells one. Our body cells also can able to produce the antigen, but this antigen is inside our cell. It is not a foreign cell that can affect the infection one. This cell antigen is good to us. Or it not say it good to us. It is does not have any effect toward the infection, will not get us sick one, but it is also as a antigen. So if the development of B cell and T cell containing certain got problem sikit, ada problem, then this B cell and T cell will attack our body. It will attack our body. So in this concept self and non-self, you need to know that how our immune cell recognize self and non-self. Self meaning is self antigen. They will know. Or B cell, they akan pergi not only B cell and any leukocyte like basophil, neutrophil, macrophage too. They will try to recognize. Or if this is self antigen, they will not engulf it. They will not attack it. They will only attack the non-self antigen only. So that is why that you need to learn about the self and non-self. So the lymphocyte in our body have the ability to detect any foreign substance incompatible with it and trigger an immune response by producing antibody. So this is general condition lah. If there are any foreign substance, any foreign antigen inside our cell, this adaptive immune response will be triggered. So the foreign substance is considered a non-self substance or antigen lah. So any antigen, foreign antigen, is recalled as non-self because it does not our body own produce one. If a foreign substance is compatible to our body cell, then no immune response is triggered, so that the foreign substance is said to be a self substance and is not rejected. So if the foreign substance like the self antigen is compatible to our body cell, meaning that it actually good to our body cell or no effect at all. No, it will not kill our body cell and just maintain at there only. So no immune response will be triggered and no adaptive immune response. So that this one we call as self substance. And then our immune cell like lymphocyte will not trigger any immune response. It will not activate. The B cell and T cell are not activating. Okay, so ability to recognize self and non-self is known as self-tolerance. So the cell, our immune cell is very smart one. It is very smart one. It will know this is our kawan. Ini kawan saya. Walaupun dia nampak macam pelik atau nampak jahat, tapi dia tahu ini kawan saya. Saya tak akan bunuh awak. Tapi kalau orang tu non-self tu, walaupun awak nampak sangat kacak, itu tak ada problem punya, nampak tak ada problem, tapi dia still akan bunuh awak. Because they have the mechanism to determine the self and non-self we known as self-tolerance. So if the immune cell is unable to recognize self, and autoimmunity will be occurred resulting the autoimmunity. So this is the fourth topic lah. So if our immune cell cannot recognize, sepatutnya kawan, tapi saya tak kisah, saya bunuh awak juga. So you will cause autoimmune disease lah. Kalau dia tak kisah apa-apa je, tengok je, bunuh je. Tengok puno. Tak perlu buat pengiktirafan punya. Okay. So establishing self-tolerance is very complicated. Actually, it is complicated system that you need to know. You don't need to know. You just want to know that it actually involves the both of the elimination of immune cell that can react self-antigen and active inhibition of immune response against self-protein. Okay. So now you need to know how the immune cells can recognize this, and this is self. This A cell is kawan saya. Ini B bukan kawan saya. Macam mana? MHC. Major histocompatibility complex protein. MHC. So you still remember the development during the development in immunity, during the activation of your helper T cell, the macrophage will engulf the pathogen, right? And then it was display the antigen with the MHC class 2. Similar to human immune response, your B cell will also engulf the antigen and display the antigen with MHC class 2. So your helper T cell, your macrophage and your B cells display MHC class 2 protein. And as I mentioned yesterday, all the cells in all the nucleated cells, so in our body or any body cells, will express MHC class 1. So MHC will play the role in recognizing self and non-self. So they will recognize MHC, especially MHC class 1. They will recognize MHC class 1. So that immune cell, when the immune cells, dah pergi jalan, if the immune cells, they found that MHC class 1 and MHC class 1, they will not attack it. Because MHC class 1 is the cell that should be occur in our body. They will not attack it. Dia akan attack punya. They will not attack it if there are no antigen. If there are antigen, then they will recognize it, attack it for cytotoxicity cell. Only cytotoxicity cell can attack it only. Attack the MHC class 1. Helper T cell and the B cell, they will not attack the MHC class 1. So MHC play the role in recognizing self and non-self, especially MHC class 1. So as we know that MHC class 1 express glycoprotein on the surface, nearly all nucleated cells play the role in presentation of endogenous peptide antigen to endotoxic T cell. Okay, so MHC plus 2 express glycoprotein on the surface of your antigen presenting cell like the macrophage, and also B cell play the role in presentation of endogenous peptide antigen to helper T cell. So MHC plays the role in recognition of cell and non-cell, as MHC consists of non-classical region which is unique for every different individual. So if you in general, you see this diagram in general, I just want to tell you for this one at the red color one. Red color. Not red color. You see the red color one. Sorry, red color one. So these are the unique region to to let the cells can recognize either it is self and non-self, because every individual are different. This one later you will learn about organ transplant. So in your syllabus, you need to know the concept cell and non-cell and relate it to organ transplant. So in general, you see movie, drama. If you want to do organ transplant, you need to do a test. You need to do a detection test, right? You need to do whether this organ, the donor organ, is compatible to you or not. Compatible test. You need to test first, and then the test they use is to test the genetic consequence of your MHC. Okay, so in general, you need to know that in general, in our body system, our immune cell will not attack the normal cell that containing MHC class 1. Because every individual, although they are containing MHC class 1, but the genetic sequence are totally different. Then because they are the non-classical region, which is totally different one. The genetic sequence are different. So, although me and you containing MHC class 1, but if I donate the organ to you, you have the possibility to reject out my organ 1. Although you and me containing MHC class 1. Why? Because this my MHC class 1 genetic sequence and yours MHC class 1 genetic sequence are different. So when they are the difference, yours immune response, immune cell will go to detect this organ is considered as non-cell. So it will attack your organ, attack that organ because the immune cell recognize this organ is non-cell because it does not containing the MHC class 1 that your self original have. So this is why MHC play the role in self and non-self condition. Because your immune cell only can know based on the MHC display on the cell only. Memang kena sama-sama sebiji baru dia tahu ini kawan saya. Kalau ada sedikit perubahan je ataupun perubahan besar, then although awak ada MHC class 1 yang sama structure dia, tapi oleh kerana genetic sequence are different, so ada ubah sikit-sikit, so your immune cell pun akan attack you punya. Dia akan attack punya. So when you relate to organ transplant, so that is why that you want you to relate to organ transplant. And that is why that you need to do compatibility test during organ transplant. Not say that if I want donate, then I can donate. No, you need to do many tests one. That's why, and why that we got rejection. So, okay, so organ transplant. So we know that there are 4 types of organ transplant. So autograft is the self tissue transfer from one body side to another in the same individual type. For like maybe for example is when you transfer when you are burn your hand, your skin of your hand is burnt. So normally your the skin doctor specialized doctor will take your skin on your leg and replace to the skin on your hand. So this one is we call as autograft because it still under the same organism, same individual, not same organism, same individual. You just transfer from different place only, like from leg skin to your hand skin like that only. So this one autograft, you will no organ rejection, but no organ rejection will occur, because still same MHC class 1, right? The genetic sequence are totally same, so there are no any organ rejection lah. So no organ rejection will occur. And isograft is occur when your organ is transferred between genetically identical individuals. So normally this one is identical twin lah. Not non-identical twin, identical twin only. So meaning that why identical twin? So isograft is where the organ transplant between the identical twins. And then this one is also don't have any rejection one because identical twin, both of them containing almost similar genetic sequence, almost same. They just only have slightly different only. Compared to others individual, compared to totally different individual like you and your brother, or you and your sister, or you and your parents, like that. If like that, still got big difference. But if identical twins, the genetic sequence almost same. So this one is also does not have the rejection one. But in real situation, in the nowadays medical surgery, medical technologies, allograft is the main that we need to use, because it is different member. like me want to donate my organ to you, this one is called as allograft. Not only me and you, like your mother need to donate your organ to you, also considered as allograft, because it is totally different member as well. Whatever you are, the parents ke, ada bapa ke, ibu ke, grandmother ke, grandpapa ke, ataupun adik-adik ke, sibling ke, semua tu we call as allograft. And then another one, xenograft. Xenograft is different species as well. Like the nowadays technology is big lah. They use they transport organ transplant using the pig, or if not mistaken, pig kidney. Using pig kidney transplant to the human one. This one is xenograft, but this technology still under the clinical test. It is not very common yet. Not common yet to all the public now. It is still under the clinical test, right? Test. And also there are many ethical problems yet. So not every country, not every religion can accept this kind of transplant. But allograft are nowadays that well developed. Okay. So rejection in allograft. So allograft confirm have rejection. There are no such thing say that allograft no rejection as well. Possibility they will have rejection. So this is the rejection. So there are actually okay, so if you see this autograft, so this autograft, you can see there are no rejection, right? But if you see this one allograft, when the this skin, if you see this skin, they receive the new skin, and then after that, after few days, especially after 1 week, inflammation will occur. Meaning that your immune cell, your white blood cell start to attack this organ, starting to attack this skin already, because they think that your immune cell think that this is non-self. Ini bukan body, bukan komponen yang badan awak sepatutnya ada. Dia anggap ini adalah bakteria, ini adalah cancer, or ini adalah virus. So dia akan attack. So when you attack, then you will get the more worst situation lah. Inflammation will occur lah. Another one, this one is more another rejection. This one is more rapidly occur. Okay, so this one I just want to show you how rejection occur due to your leukocyte. Your inflammation will be occur. Okay, so your tissue that share sufficient antigenic similarity, allowing transfer without immunological rejection, are said to be histocompatible. So normally this one is only occur in identical twin. And another one is tissue that displays significant antigenic difference, meaning that your antigen are different as well. Your antigen on your surface are totally different, meaning that your MHC class 1 is different, is we call as histoincompatible. Histoincompatible meaning it is incompatibility lah, and typically induce an immune response that lead to tissue rejection. So normally this one is occur only allograft and also xenograft lah, but xenograft because to confirm got rejection lah, xenograft. Allograft still have low possibility that does not have rejection. But xenograft, if you not do a genetic sequence like that, you just directly transfer, confirm 100% can say got rejection. But unless you change the genetic sequence of certain of the donor organ, then you can reduce the rejection rate. Okay, now we go to rejection mechanism. So rejection occur in stage and can be caused by both humoral and cell-mediated immune response to allo-antigen MHC expressed on the self of the graft. So there are two phases: sensitization phase and also effector stage. So in sensitization phase, normally is occur very short and rapidly after transplantation occur. So meaning that when you finish up your organ transplant, then this phase will directly occur. So meaning that this phase, your lymphocyte, your B cell and T cell will proliferate at the organ transplant area, your donor organ at there, and then to respond the immune response. And another one is effector stage, which the immune cell destruction of the graft take place. Okay, so this is all about the rejection mechanism. Okay, under the same topic, you will need to learn about HIV infection. Okay, so HIV infection. Okay. Okay. Okay, so I think HIV virus, I think I don't need to explain many HIV virus. I think everyone should be know it. So when the people get infected HIV, they will become AIDS. So I think this one, the common things I don't need to explain, but you now in your syllabus you need to know the mechanism of infection of HIV, because everyone know HIV virus is already appear very years already, 1,000 years, but until now there are still no ways to kill this virus. There are no any treatment. For nowadays, the treatment is just reduce the symptom only, reduce the virus, not infect the cells only. No, there are still no way, any vaccine or no any medicine can kill this virus. For now lah, until now. In future, maybe you guys do a research, medical research, then you guys found that got certain drug or medicine can kill the HIV virus. If you are the one can found this, wow. I can say you will be the good, you will be the highly contribution people in the world. Semua orang akan macam itu, orang famous lah, orang popular lah. Okay. So you need to know HIV virus. This is the general structure of HIV virus. So HIV virus is the single-stranded RNA virus. It is a single-stranded RNA virus which envelope virus. You can imagine it is like the COVID-19. The shape is look like COVID-19. The shape is still look like the COVID-19. Right? So I think the COVID-19 punya gambar semua orang tengok banyak kali kan. So you can imagine it like that. So it is an envelope virus, virus, and also the genome of the virus is single-stranded RNA. It is an RNA virus. And then that in their envelope they containing the spike protein. This one is we call as spike protein. But yang ni PGP 122 ini awak tak perlu tahu lah. You need to know that this one is the spike protein. And then inside the virus they containing the special enzyme we call as reverse transcriptase. This is the enzyme very important during the infection, where this enzyme does not contain it in our body one. Our human body will not have this kind of enzyme one, because reverse transcriptase, because you are now in SEM 2, you are not entering SEM 3 yet. So very difficult to tell to you. So I just tell you in general, if you learn in primary school or secondary school about the virus, you know that virus actually is considered as not considered as organism one. It is not microorganism one. We cannot say that virus is a microorganism, and we cannot also say that virus is dead organism, because virus containing two alive and dead characteristic. Because virus does not containing any organelles one. It only contain viral genome and also certain enzyme only. So virus only can survive. It need to based on the host cell one. It only can survive when they penetrate into host cell. They cannot survive alone one. Tak boleh, dia tak boleh hidup secara individu punya. Tak boleh. Dia kena ada satu, dia kena masuk sel baru boleh survive. Baru boleh replicate. If not, the virus cannot, they cannot survive one. They cannot replicate lah. In general, they cannot replicate individually. They need to go to the host cell, infect the host cell, and use the information inside the host cell to survive. So actually, this topic, I think it should be in chapter Sem 3 because it more about on Sem 3 topic 1. But never mind, I try to explain it. Okay, so this one is the mechanism. Normally, it will involve the central dogma theory that you learn in Sem 3. So, in semester 1, you will still remember the function of mRNA, function the nucleic acid. We know that in our human, we will bombard the central dogma mechanism, right? Where your nucleus containing the genetic information, your DNA as the carry genetic information, and then this DNA data will be transcribed into mRNA, right? mRNA, and then this mRNA later will come out from the nucleus and translate to become protein. This one is the general concept. I think you learn in Sem 1 lah. Okay, I assume you already learn in Sem 1 this one. So the virus, as I mentioned before, virus cannot survive alone one. They cannot replicate alone one. They require the component inside our cell one to replicate. And then another reason I need to tell you why your HIV virus is very dangerous virus if you do not get the treatment, because HIV virus very smart. It will not attack the normal cell one, not like COVID-19. COVID-19 attack what? Attack our lung, right? It attack our lung. HIV virus very smart. It attack helper T cell. Wah, sangat clever lah. It's very clever. It attack your immune system first, and it attack your initial immune system helper T cell directly. You know, if you still know that the immunity development, the first step, initial step is activation of helper T cell. You need to activate your helper T cell first, then followed by you have cell-mediated immune response and also humoral immune response. But HIV are very smart. They directly kill helper T cell. So once your helper T cell already killed, so dah mati kan, helper T cell dah mati. Mula-mula punya askar dah mati. So your B cell and your cytotoxic T cell cannot receive any signal. So this B cell and cytotoxic T cell are not activated. They are just stay at there only. Tak buat apa-apa langsung. So at the end, the patient AIDS not die due to HIV virus. They die due to the weakened immune system, because their immune system now is very low and very weak. Any type of pathogen, any sick, like if in general normal, like we get the flu, we normally just sick 3 days, 4 days, then we will get well soon and we eat medicine or eat antibiotic. But in AIDS patient, if they do not get the treatment, they just bear macam tu je. Then as the long time, your immune system will become weakened, and once you are exposed to any type of pathogen je, due to there are no immune response, no adaptive immune response, so you will die immediately. You will get very serious disease, and even though you will die more easily. If the pathogen are very strong, like COVID-19 like COVID-19 you still remember in COVID-19, even though healthy person also have the possibility to get into ICU. So this kind of AIDS, you imagine helper T cell already tak ada, already AIDS, another virus COVID-19 masuk lagi. Memang itu gila lah orang tu. So kena tahu lah. So So that is why of the reason why HIV virus is very dangerous virus, and also can cause very dangerous disease and very severe disease. And normally they die not because of HIV virus, they die because of weak immune system, and then weak immune system and due to the infection, any infection. Okay, so now you need to know about the mechanism of the virus. So in general, your HIV virus basically the spike protein GP120 is the spike protein. It will directly bind toward your CD4 accessory protein on the helper T cell. CD4 protein on your helper T cell. Okay, after that they will release the viral genome. So your nucleic acid containing the viral genome and the enzyme we call as reverse transcriptase will enter into the cell. So it will release all the component in the virus, it will release into it. So the main component is viral genome and also reverse transcriptase. So once the viral genome and enzyme are released following removal of core protein. So this core protein, this surface protein, envelope protein will remove lah. It will release out. But and now your helper T cell is already infected by HIV virus. Okay, now your virus require reverse transcriptase. Okay, if you still remember, in central dogma, our human central dogma is you need to use DNA become mRNA and then become protein. Okay, now this virus, because RNA virus, they cannot directly become mRNA one. Double A. Tak dapat. So this virus need to use the reverse transcriptase to form RNA DNA hybrid. To form another DNA, because reverse transcriptase function is to change RNA to DNA, terbalik punya. Like in human, we change DNA to mRNA in human, but this enzyme is a reversible reaction that human does not have. So it will catalyze the mRNA to form RNA DNA hybrid, and then after that your original RNA will be the template and is partially degraded by the ribonuclease enzyme, followed by synthesis of second DNA strand to form double-stranded DNA. Now your DNA is your virus is forming viral double-stranded DNA similar to human. Because they need to change to DNA first. Because our human enzyme, semua human enzyme ni tak dapat change single-stranded RNA kepada mRNA one. No, there are no such thing. There are no any mechanism like that. So viruses are very clever. They are sangat pandai. Mereka sangat pandai. Kerana mereka tahu lah. Oh, kerana human tak dapat. Tapi saya nak guna human punya enzyme. Kerana yang awak kena tahu, virus dia tak dapat replicate own self kan. Dia kena guna hot cell punya enzyme to replicate. So they want to replicate. You want to use the mechanism of the cell so that they need to change to DNA first. So once they already change to DNA, this DNA later it will translocate into the nucleus and integrate into the host chromosome DNA. Meaning ini DNA dia akan pergi nucleus, and then this DNA akan campur dengan kami punya DNA. Campur je. Integrate lah, we call as integrate. Dia macam campur je. So now your cells containing viral DNA already. Viral HIV DNA. So after that, now interesting is now your cell in normal, macam normal, your cells still can undergo transcribe. So mRNA, the transcription still occur to form mRNA, but now due to your DNA, your cell, your DNA containing the viral DNA already, but your cell tak tahu. Dia tak tahu lah. Dia hanya tahu ini DNA kan. Oh, ini DNA. So dia terus je lah. RNA polymerase terus pergi je, and then transcribe and to form mRNA. So now your mRNA is viral mRNA. Ini mRNA bukan mRNA yang biasa. Ini mRNA ada viral mRNA. Okay, then this mRNA keluar lagi. So macam biasa keluar lagi and then undergo translation. So virus pun tak tahu. Your tRNA pun tak tahu. Oh, ini mRNA kan. Oh, translate lah. So translate to form protein, and this protein is viral protein. Ini viral protein viral protein. So this viral protein will assemble together to form the membrane of the cell. So you need to know that the membrane of the virus is made up of glycoprotein, is one type of protein. So this protein will assemble together to form this viral protein. And then there are the RNA exported to the cytoplasm, and then this membrane bud out, forming the viral envelope, and finally new HIV virus release. And these cells is already infected, and it will die at the short time, and this cell now cannot function anymore. Helper T cell cannot function anymore because your nucleus, your genetic content is totally different now. It already totally different. It already undergo the very serious mutation. So your helper T cell is already cannot function anymore. So this helper T cell only can do one function only. Copy the HIV virus. Copy banyak, and then this HIV virus affect another helper T cell again. So at the end, if you not control it, your helper T cell will die anyway. Akan mati, semua akan mati. In the end, your body does not containing any helper T cell anymore. So very dangerous lah. Sangat bahaya lah. Okay, so this is a mechanism about the HIV virus. Okay, so another lah explanation about this. Okay, so I think I don't need to explain why. Okay, next is about the last topic today lah. So today this is the last topic, which is a immune disorder. So this one is that I just explain about concept self and non-self, where your immune cell actually can able to detect self and non-self. And in terms of organ transplant, due to the different sequence of MHC, so that the organ, donor organ able to be destroyed, to be attacked by your self immune response, the organ transplant. But certain day we call as autoimmune disease or autoimmunity, where your cell tak ada organ transplant pun, but your cell is already got something problem. Tak kisah dia tak dapat recognize, it already cannot recognize. Organ transplant, they still can recognize. They recognize actually normal one. Just that awak ambil satu benda yang memang tak sama punya, so that is why your immune cell attack it, but it's still normal condition. Your immune cell is normal, function normally je. But this one is autoimmunity disease, is actually your immune cell is already not function normally. It already attack your self tissue. Meaning that your immune cell, your lymphocyte are able to recognize self and non-self anymore. That bunuh je, apa-apa je. So in the real world, in the medical nowadays, there are many types of autoimmunity disease. But in your STPM syllabus, you only learn about this one, systemic lupus erythematosus, SLE. So this is the disease. So I think disease, one of the famous autoimmune disease now in the world. Because the probability of get this disease is quite high compared to other lah. If not mistaken, is 1/100. And then this disease especially will affect women. Women are easy to get this disease compared to women. The probability, if not mistaken, is 1 to 10 lah. Meaning that 10 women, 10 we've got 10 women get this disease, only 1 man will get this disease. Dalam satu, itulah probability. And then in the real world, 1 to 100, meaning that in 1,000 population, got 1 person will get this disease. 1 to 1,000. And then if ratio between men and women is 1 to 10, meaning that got 10 women get this disease, only 1 man will get this disease. So this disease is very famous in women lah. Okay. So this is the general mechanism how your immune cell attack your self tissue. So this is entire your body one. It will entire body. This disease is your antibody. The main antibody should be protect you. But now because your autoantibody are formed. Autoantibody meaning that this antibody is already conformed will attack self antigen. It will directly attack yourself. Okay. So so normally this antibody is against your DNA. You're against your histone, any nucleus component. It will attack your nucleus one. Kalau dia tengok nucleus tu, dia attack je. So you see this diagram. So this one is B cell. So you already, because due to your autoimmune disease, so got something problem. So your B cell now secret antibody, and this antibody is will attack your self tissue. So this B cell now is abnormal. So your circulating B cell will bind self antigen released from injured cell. So when you are injured, you will release self antigen one. So this self antigen actually does not have any effect too. Tak ada apa-apa effect yang buruk kepada badan awak kerana you just injured je. But your B cell, so in normal situation, your T cell and B cell will not take any action one. Tak akan buat any action one. But because your B cell and T cell are abnormal one, so your T cell, your helper T cell will also bind this antigen, and your B cell also bind this antigen. So when they bind this antigen, this B cell is activate. Should be not activate, but now it activate. So when it activate, so the helper T cell activate this B cell. So this B cell is activate. So the B cell starting to secrete plasma cell, and then this antibody will attack this antigen. So this antibody dia akan pergi. So kalau dia pergi ke sini, ke tempat yang darah yang dah injured, go to the place at the injured, this antibody will trigger the inflammation reaction. It will trigger the antibody reaction lah. Semua tu akan trigger. So it will destroy all the cell. So at the site of injury, self antigen and specific antibody will initiate an inflammatory response, causing more cell injury. Dia ada bind antigen ni kerana antibody ni dia akan pergi cari, and then dia found that dekat sini ada banyak cell antigen. So dia akan bind dekat sini, cause inflammation, and then your cell akan destroy lagi. Inflammation in normal situation is good. Dia baik, tapi dekat sini dia tak good. Dia akan destroy lagi, and it will kill the cell more and more. Dia akan bunuh sel yang lebih banyak. Sepatutnya hanya satu tempat je, tapi sekarang due to this antibody and function autoantibody, dia akan bunuh lagi banyak sel lagi. So it will have the positive feedback effect lah. So why? Because your macrophage and your and your macrophage and your neutrophil will trigger phagocytosis to engulf this all antibody, right? So more B cell will bind cell antigen, amplifying the cell cycle of tissue damage. So you will get the cycle. So if you not kill that, you will not stop the antibody, then this process will not stop. Dia akan bunuh banyak normal cell lagi, and then causing inflammation again and again. Jadilah, and then ini B cell lagi, abnormal cell B cell lagi trigger again and again. Dia macam recycle lah. You cannot stop it. If you do not use any medicine, you do not use any drug to stop this antibody action, then you will get more serious effect. And then after that, the main serious effect is you will get type 3 hypersensitivity reaction, where your blood vessel will be destroyed. It will destroy your blood vessel. So this complement activation will generate member attack complex, and complement treatment will damage your wall of blood vessel, resulting vasculitis and also glomerular nephritis. So this is the inflammation of your blood vessel. You know that you already destroy your blood, you destroy your cell. So even your blood cell, your blood tissue, your blood vessel also containing endothelial cell, right? This cell also can be attacked by this antibody. So once it attack this blood vessel, attack the endothelial cell, it will cause vasculitis, inflammation of the blood vessel, and also your glomerular also can be inflamed. So when you excessive again, if you not control again, leading to neutrophil aggregation and attachment to the vascular endothelium. And finally, the number of your circulating neutrophil will be decreased, and occlusion of various small blood vessels will develop, which can lead to widespread of tissue damage. Because inflammation is still occur, and then it will spread your tissue damage, so that you will see this kind of people. So the symptom of this SLE patient, you see this, normally you will see the patient muka dia sangat merah punya, we call as red rush. Dia bukan macam panas orang yang sangat awak pergi panas sangat awak muka awak jadi merah, bukan. Ini in general situation dia pun akan jadi merah punya. And then dia due to the inflammation of blood vessel, awak boleh tengok dia tangan dia jadi bengkak punya. You see the tangan dia sangat besar punya. Ada banyak symptom lagi lah. So ini all the symptom that may be possibility to have. Okay, so so that's all for this chapter, and that's all for today class. So any question for today class? I think no. Okay, so today class only 1 hour. Okay, so now to this semester I already complete the 6th chapter already. So next week, I think next Tuesday, it will be the last class for this semester, because infection disease chapter 13 is very short topic and very easy topic only. So I think I can finish up in 2 hours. Okay, so next week will be the last class lah. So I hopefully everyone can join again last class for this semester. So okay, I think no question. So that's all for today. So thank you for your joining, and have a nice space. Good night and good days. Bye.